So the title of my talk was to be genome editing for animal health, but obviously health is many different aspects, whether that's stress resistance, heat resistance. So today we're going to talk about disease resistance. And the primary example I'm going to talk about are the PRRS-resistant pigs, because that is what we've done and that's what is the most evolved in the approval process. However, as Simon has already highlighted, one of the biggest issues that we have is finding targets. And where we can take our inspiration for genome engineering for disease resistance is from GWAS studies. We can look at inter and intraspecies variants, which we have discussed a lot for things like African swine fever, where we have wild suets that are resilient to the disease. But the by far most interesting part for me is the in vitro host pathogen studies, and I'll explain to you why. And again, here we have two different avenues. We can go for genome-wide screens, usually a long shot, or hypothesis-driven approaches. So the major problem that we have when we want to look for disease resistance in a population is that we see these piglets and we may associate what they are behind it. So we can measure how much virus they release, and we say the low virus releasing pig is resilient. However, if that is just in a population, this pig may have not been exposed to the same amount of virus to begin with, so it has produced less virus. So we only see the outcome of very diverse disease. So the only solution we have to that is actually challenging thousands of animals with the same amount in the same conditions, which is too expensive, not ethical, and bottom line, impossible to do. So we have to take our inspirations from other places. In saying this, genome-wide association studies have managed to produce resistant animals in pigs. Good examples are the F18 and the F4 enterotoxigenic E. coli resistant pigs, which are to find in this resistant pool. And those are probably the easiest animal to find in a genetic pool, but none of the other ones. So today we're going to talk about PRRS-resistant pigs, and the background to this doesn't come from the animals itself, it comes from in vitro studies. So for those who don't know PRRS or think that is what the noise that a cat makes, porcine reproductive and respiratory syndrome, it is a mouthful, so PRRS is much easier. It is a viral infection in pigs that causes respiratory distress, fever, and inappetence in animals of all sizes, but it's particularly important in suckling piglets, where it can lead to up to 100% mortality, depending on strain, but also in pregnant sows, where it can cause complete abortion and death of a fetus in utero. So that is a strong economic impact. It leads to animals suffering from disease, but more importantly for the production aspects, it leads to a loss of animals, growth, and eventually it's food waste in the production chain. What is also very important about this virus is that it infects macrophages. So the first line of defense of the immune system, it manipulates them. And studies from Europe have shown that the antibiotics use in farms that are positive for PRRS is at least double, up to 5-fold more than farms that are negative for PRRS. So the secondary bacterial and pathogen infections are absolutely important for this disease. It is a global problem. It's a pan-zootic that costs us billions of dollars around the globe. And until the recent outbreak of African swine fever, this was the one to beat for the pork industry. And here is where we go into in vitro research. We look at how this virus interacts with its host, and that's what my daily bread and butter is. I'm more on the virology side of this, but also then translating this, of course. And what we're looking at is actually this small part of the virus's life cycle, when the virus has to escape the endosome. Otherwise, actually, this is a destructive path. So the good thing is, also from kind of an evolution perspective, The virus is already inside the cell, and if it would continue on this path and couldn't escape, it would be destroyed by the cell because it wants to reuse the building blocks of this virus. So that makes it very interesting. And we know that the receptor in here is a protein called CD163. It is a protein that has a structure that looks like 9 beads on a string, and only bead number 5 interacts with the PRRS virus. However, here's the first problem that we face when we want to do genome editing. Every protein that we want to edit has a biological function. CD163 has many. It is a hemoglobin haptoglobin scavenger. It is important for red blood cell development. It binds bacteria and presents them to the immune system. It is important as a soluble version in orchestrating our immune system. But surprisingly, bead number 5 is still green. It hasn't got any biological function associated to it. But for some reason, evolution kept it there. But we know that there's evolutionary duplication of a CD163-like protein that has 12 domains. So somewhat there is a little bit of wiggle room in evolution here. So we can just try and remove domain number 5. Ideally for us, the genetic locus of CD163 domain 5 was perfect because it's a single exon that encodes it, and removing that exon keeps the whole gene in frame. So the solution was simple. Target genome editors at either side, remove domain number 5, and you get strings on a bead that just are 8 instead of 9. Structural prediction was predicting that this was going to be correct, and indeed it was. After lots of characterization in vitro, monitoring biological functions such as hemoglobin haptoglobin scavenging, the animals were then exposed to the virus. And what we can see is the bottom line, that these pigs neither get infected nor do they seroconvert. So we don't even seem to see a low level of viral replication in these animals. Now, what we've done since is taking this, and that is very, very dull, taking this a step further. So we're going back to the genetic locus, and we understand quite a lot about how genetic regulation works. And if we look at the exon, the beginning of the exon, number 7, we know that this AG sequence at the beginning of an exon is absolutely crucial. So we can actually make minor modifications to this. We can actually change single bases to get the same effect. So we can target a guide here and just do a random change and a random short deletion, or we can use an HDR template to make a single change, for instance, to this G. Or last thing is we can use genome base editors, which don't cut the double strand and always give us the same outcome in removing this G. Sadly for us, we tried the base editors, and the base editors at this position are predicted to be really poor at 0.4% efficacy. And the conclusion was we cannot edit this well enough. So we had to kind of go for the random deletion or the HDR template. And one of the options we have is an 11 base pair deletion pig. And lo and behold, this has the same phenotype and the same reaction to viral challenge in vitro, but a minimal change. And we have since made this edit with an HDR template to only change the G. And once again, we're at the same phenotype with a slightly different genotype. But that gives us an interesting thought process. If a single base change is sufficient to give us a PRRS-resistant pig, logic tells us with mutation rate, that pig is probably out there. But first, we'll get back to that discussion. What are the opportunities for this pig? Well, improved animal welfare and improved production. We don't have secondary bacterial infections anymore. We use less antibiotics. But it's also an interesting concept to deal with this in a mixed population where we have wild type animals and animals that work like a natural ring vaccination, where you can actually start with phasing out PRRS infections by improving a biosecurity ring with resistant animals. So why do we need genome editing if these animals might be out there? Well, the first thing is that we might have a population that has a trait, but it's just not there in enough prevalence and we risk inbreeding. It could be that this trait is present in an indigenous breed that is just not productive enough to get back to our high producing population. Or again, if we look at African swine fever as an example, we have the possibility that we cannot crossbreed and integration, natural integration is not possible. So we talk about cisgenesis more or less, or we have in vitro results. And in vitro results are amazing because in vitro, we can challenge one cell with thousands of viruses, thousands of conditions. So it makes finding SNPs to some extent or finding traits much easier, but they're not always editable because our genes of interest have other functions. So what we end with when we genome edit for disease resistance can be different things. One of the options is we end up with a tolerant animal, and we have done that with natural breeding over decades. What we have ended up though, especially when we look at chicken, is that we have a very high pathogen burden, but not a high disease burden. But that gives us the risk of both evolution of the pathogen, but also kind of tipping over the balance and getting more problems once we basically tip over the stress limits and get disease taking hold. We could end up with resilient animals, and I think in many cases we have to accept that not all genome edit will lead to complete resistance. But what we have to keep in mind that life attenuated vaccines may show less efficacy in animals like that. And we'll get to that. Of course, the resistant animals are the golden goal, and PRRS resistant animals seem to be. Another experiment that we've done in vitro is take primary cells from the edited and the wild type animals and try and push evolution in vitro. So challenging them with many different biostrains, co-culturing wild type and edited cells at different ratios and passaging over many passages which mimic natural infection passages. And we've not seen any evolution. So what do I think are, obviously I see evident benefits of genome editing for disease resistance, and I think it's very difficult not to see them. I just think finding the targets is probably not the easiest thing. So we should see genome editing as one of the tools in our belt. And especially when we talk about resilience or tolerance traits, it is good to think about developing other methodology alongside. We have new vaccine technology that is not very broadly used in animals, which is RNA vaccines or amplifying RNA vaccines, affordable antivirals, basically not touched in animal biotechnology, but also the obvious antimicrobial alternatives. But the other thing I also want to go back to is, what do we regulate? Because if we see these different methods by which we can achieve PRRS resistance, what do we regulate? Is an HDR template integrating foreign DNA, even if it just leads to one phenotypical base change? All of these questions that we're faced with when regulating this technology. And the other thing I want to leave you with is that whilst it's difficult to find targets that are in the genome to edit, integrating antiviral strategies into a genome as a transgene is a very viable option, especially when we look at things like CRISPR or these DNA, RNA targeting tools. So there are lots of opportunities here that we shouldn't categorically exclude. So thank you to my group. These are the major funders for this project, and this is our other funders that my group receives funding from. And because it's much easier to finish with photos, that's the theme in beautiful Scottish weather. And these are our international collaboration partners in the Philippines. Thank you. Not on genome editing, but on other projects.